Hi, and welcome to your notes on the ear. Let's see, your ear has two functions, hearing and also your balance. The receptors in your ear are called mechanoreceptors because they respond literally and to physical forces and the movement of hairs and fluids in your body. That's why they're called mechanoreceptors. Okay, and the equilibrium part of your ear has different receptors than the hearing part, so those are separate organs actually. So your ear is divided into these three areas: your external ear, your middle ear, and your inner ear, right? Most of the important stuff happens in your inner ear. The external ear is called the pinna, or the external auditory, or the auricle, sorry, not the external auditory canal. This is the external auditory canal here, also part of your outer ear. The auricle here has an interesting shape, and it is used in many mammals to draw wavelengths of hearing into the external auditory canal, although I think the idea now is that in humans, because our ear lobes are so small, that it doesn't serve much of a function, but that is why our ears are shaped the way that they are. Your external auditory canal, if you recall from our study of the skull, the skeleton, you have an external auditory nidus. That's what we're talking about here. Remember, it's above the styloid process and the mastoid process in your temporal bone, right behind the xiphoid process and the hole for your eye, the orbital. So it's lined with skin. It's about 1 inch long. Oops, I need my pen. 1 inch long, and it's about 1/4 inch in diameter, inch wide. It has ceruminous, that's how you pronounce this word, ceruminous, or these are your wax glands. They produce cerumen. These are, I'm going to add a little bit to this. These are modified apocrine glands. Apocrine. So remember, you had apocrine sweat glands that were specialized sweat glands in your armpits and your groin. These are also modified sweat and oil glands, and they contain a thick substance which helps to just protect physically the skin of your ear, and also they have been found to contain lysozyme, or basically antimicrobial properties. Okay. Your external auditory canal, the category, your outer ear ends at your eardrum, which is your tympanic membrane. This is your eardrum. And past your eardrum becomes your middle ear. Your middle ear is filled with air. It's only involved in the sense of hearing, that meaning your inner ear has two functions, hearing and equilibrium. Your middle ear is only involved with your sense of hearing. This is a dumb slide. I don't like it, so we're going to alter it a little bit. There are two openings. Love my pen here. Two openings associated with your inner ear. Okay, those are called, so you can add this, and there's a space under there under the picture, you can add it. So your inner ear has a vestibule, and there are two holes, basically. There's one called the oval window. The oval window, this is associated with one of the bones of your inner ear, one of your ossicles. And the other one is called the round window, and we'll study that when we get to your inner ear. But these are openings, little holes essentially. Okay, and then you have one tube in each ear. You have an auditory tube. The opening. What this line here is saying is that the pathway from the external ear to your middle ear is covered by your tympanic membrane. Where air does enter and exit is through the auditory tube, which you can see in your picture. And the auditory tube is how you equalize pressure. Atmospheric pressure, essentially, when you go to Bishop, but otherwise your ears hurt because the pressure increases. And essentially what's happening is that you don't actually get even vibration on your tympanic membrane without having the air pressure be the same on both sides. So that's why it gets harder to hear, and sometimes you get ear aches, and so your ears pop is how that tympanic membrane adjusts itself. The tube, unless your equalizing pressure is collapsed, there's an interesting homeostatic imbalance in your textbook about ear infections in children, and supposedly this auditory tube is more horizontal in children, so it doesn't drain well, and sometimes kids have to get a little tube put in their ear. This is called a myringotomy, and they stick it in your eardrum, and that lets the pus that builds up, inflammation inside your ear, drain out. So also in your middle ear you have 3 bones. And these are the bones, these are the smallest bones in your body. They're called the malleus, the incus, and the stapes. The hammer, the anvil, and the stirrup, more affectionately called, and they really do look like that if you look at your picture. And they are what carry the vibration from your eardrum into the middle ear. So the actions of these 3 bones, essentially their functional structure, cause amplification of the sound waves that pass into the eardrum. So here's the eardrum, and it's causing vibration, and the hammer vibrates. And then the way that the anvil, the incus, is structured with the stapes, it's causing the stapes to pound on this fluid-filled vestibule here. And behind the stapes is the oval window. So that's where the sound is going to go into the inner ear, the cochlea. And just the way that this is set up amplifies that sound into this, so that the total force of sound that reaches the eardrum here over the whole area of the eardrum gets pushed into this tiny oval window, carrying all of that sound, and so that's the amplification process through the function of those 3 bones. Okay, here's where we get to the meat of how you hear. Your inner ear, also called the bony labyrinth. So it includes organs for hearing and balance. Essentially, your cochlea here and the vestibule are involved in hearing, and your semicircular canals are what's involved in balance. They are filled with another plasma-like fluid called paralymph. These three structures are actually not something that you can like take out. It's not its own organ. This thing here, it's actually just a series of bony chambers. So when your book says, when they show you this picture here, it's like taking a plaster of Paris mold and, you know, filling the chambers with plaster of Paris and then removing it, and then you can see kind of the structure. So this is all actually bone-filled chambers, and the three of them: cochlea, vestibule, and the semicircular canals. You have three semicircular canals. And we'll get to those when we talk about balance. Okay, let's start with the cochlea. The cochlea is that snail-like membrane. It is inside the cochlea. There's these two fluid-filled membranes are filled with paralymph, and then there's a third one, and the third one in the middle is called the organ of Corti, and that's the actual hearing organ, the organ of Corti. It is filled with a slightly more thicker, more gel-like fluid called endolymph. It has two membranes. It has a basilar membrane. That's like the base, so we can write base here. And the basilar membrane houses the receptors with the nerve endings that go to your cochlear nerve. On the surface of those, so those are hair cells. The things that detect, that cause the movement, so is the tectorial membrane. And this is just a gel-like membrane, and it's the thing that moves. The tectorial membrane is what moves and causes the hair cells to bend. So it moves with changes to the endolymph, and the hair cells then pick it up. And then those receptors send information to the cochlear nerve and transmits it to the auditory cortex on your temporal lobe, which is your cranial nerve number 8, 5, 6, 7, 8, the vestibulocochlear nerve that then goes to your auditory cortex. Okay, here are the pictures you needed. While you're labeling, I will point out the different structures. Here's the vestibular membranes filled with paralymph. They're just fluid that help carry that vibration. Here in the middle is this other membrane. This is your organ of Corti, your hearing. This over here is a blow-up of it. So here's the tectorial membrane that moves with the fluid and causes the receptor cells in the organ on the basilar membrane, the base membrane here, causes them to bend, and which sends information to your cochlear nerve. Okay, so these notes go with the pictures at the bottom of your page, and it's going to be easier for me to explain if I'm looking at the picture, so you can just copy these in here. The one thing that I'll add where we are here is this last bullet point about continued stimulation. And essentially what this is this happens in our brain, not in our ear, obviously, but our brain tends to adapt to noises. So, for example, if you hear the refrigerator going when you walk into a room, after like a minute, you don't hear the refrigerator anymore. So if the same stimulation is occurring to those nerve cells continuously, our brain tends to sort of set them aside and not pay attention to them anymore. And that's the adaptation. Okay, so here's what's happening with those vibrations. So this picture up here is basically taking all of the curvy part of the cochlea and spreading it all out. And what you're looking at here is this fluid membrane here, the outside, the perilymph. Those are the two perilymph-filled vestibular sacs. And then this thing in the middle here, this is going to be where your organ of Corti is. That's the endolymph-filled part. And essentially what happens is the information comes into the stapes here, and it travels like all the way up to the top of this circle thing, and then turns around and comes all the way down. Okay? And somewhere in here, all along here, you have the hair cells. They're in the basilar membrane here. So information travels up. This first part, it's called the scala vestibuli, or vestibuli, and then it travels down through the scala tympani. And depending on the frequency, so when we're looking here, down here, we're looking at frequencies. This one's a little, I don't like this picture so much, but this is one vibration, one frequency, right? It comes in, it gets amplitude by the three bones of your middle ear, and then flows through, and then over time it ends up coming out. So the sound waves come up, go down, and then they come out the round window. So in through the oval window and out through the round window, depending on the wavelength and the frequency. So high frequency, high-pitched tones, they get amplified really quickly into the basilar membrane, which is at the bottom here, and by those cochlear nerves, the hair cells. And then lower tones take longer and end up getting incorporated into the cochlear duct further down. And so this is kind of like, if you think of it, I like to think of it like a piano keys, where the different sounds are getting incorporated into your ear along the cochlear duct based on, and that is kind of how it happens, right? It hits a key, hits a vibration based on the wavelength. So that's how it works. There's a video that I posted on the class website, and it's also on my YouTube channel that you should check out, that shows really well how that all happens. But that's it for hearing. Okay, so now let's talk about equilibrium. You have two different organs for equilibrium. You actually have two types of equilibrium. Each one does a different one. So one of them is also in the vestibule of your inner ear, and the second one is in the semicircular canals of your inner ear. The static equilibrium is, oops, let me get my pen, is the one that happens in the vestibule. And your dynamic equilibrium is measured by the semicircular canals. And then the information, of course, is sent via the vestibular nerve to your cerebellum, right? Not your cerebrum, not your cerebral cortex, but just to the cerebellum. Remember, your little brain is the thing that manages your balance. So that is still part of cranial nerve. I have my pen. 8, 5, 6, 7, 8. So there's two branches to the vestibulocochlear nerve. One goes to the cochlea, that's for sound, and the other one is the vestibular branch, and that's for balance, and that just goes straight to your cerebellum. Okay, and these two images that are labeled here are down at the bottom of your picture also, so you're going to label them. Those are figures A and B at the bottom of your page there before you move on. So your static equilibrium is essentially when you are not moving. It's your response to gravity, and so it's essentially what tells us what direction is up and down. So up versus down. This is what is responsible for swimmers being able to determine which way is up to the surface. And it's how we feel basically the pull of gravity. The way this works is that in the membrane of your vestibule, you have little hair cells again that are embedded in what's called an otolithic membrane, and it has a gel substance and it has these tiny stones. These are made of calcium salts. And they're floating in the gel, and then when your head tilts in some direction against gravity, the weight of the stones pulls the gel and causes the hair cells to bend in that direction, telling you which way is the gravity is pulling you. So here's the picture of it. Here's the membrane. Within the membrane are the maculae, they're called, or maculae, which means it's Latin for spots. And so you have your little salts in the gel here, and you have your little membrane that has the hair tufts and cells and the nerves in it. And then when the salts, the weight of the salts, so the otoliths, pull on the gel and pull on the hairs and tell you which way is down. That is the maculae static equilibrium. Your dynamic equilibrium is more kind of every other kind of movement you have. Angular, rotatory movements of your head. Your book talks about twirling on the dance floor, or getting seasick while you're in a boat ride, having those receptors work overtime. So you have 3 semicircular canals. Maybe you want to write this. The 3 semicircular canals are oriented in the 3 planes of space. Okay, and so whichever way you move, there are receptors that will respond to that movement. These response receptors are called crista ampullaris. That's these things here, crista ampullaris, and they're located kind of at the entrances to the semicircular canals. You already labeled one of them. That's figures A and B at the bottom of your page there. So that's a picture of the crista ampullaris. And inside each crista ampullaris is what's called an ampulla and a cupula. And the ampulla is kind of the membrane, and the cupula is this little gel-filled part here. And it acts kind of like a little wavy cap. So if your body moves this way, the cap flows that way. And that's how it's determining the direction of the flow. There is kind of the, when you stop, you can send, like when you stop in your car, there's a backwards, reverse motion sensation. And you can kind of feel that too when you stop suddenly. If you're like in a car and you're moving at a constant rate, the receptors adapt again and stop sending information. Same thing happens if you're seasick. Eventually, these nerve fibers will just give up, and that's when you get over your seasickness, when that overworking finally gives up and it no longer keeps track of these excitatory motions of your body. So this slide, I guess, just lets you finish filling in your notes. I would add one more thing, and that is that pretty much static and dynamic equilibrium act together to keep you balanced. as well as the cerebral response of your cerebellum to your muscles and your tendons. All of those function along with these sensory nerves to control balance. So that's it for your hearing. I would encourage you to read one more page. I'm going to write it down here. Page 270 in your textbook talks about homeostatic imbalances of your hearing and equilibrium. And so it talks a little bit about deafness, and then also talks about equilibrium problems with dizziness and infection and how that's related to those symptoms. Other than that, we're all done with the ear. Thank you.